for a long time, one of the biggest dreams in medicine has been this idea of cellular reprogramming. Telling one type of cell to become a completely different one. Imagine taking a regular old skin cell and turning it back into a powerful blank slate stem cell that could repair anything in our bodies. Now, the ways we've tried this before have been, well, a bit clumsy. But what if I told you the secret to rewinding that cellular clock isn't some brand new invention? What if it's been locked inside ourselves this entire time? I mean, really think about that. What if we could hit a rewind button on a cellular level? Tell a cell to just forget its current job and go back to its factory settings, back to that state where it was young and could become literally anything. This isn't just science fiction anymore. This is the very real future of regenerative medicine. We're talking about growing new organs, repairing devastating injuries, and maybe, just maybe, even reversing the aging process itself. Okay, so here's the crucial difference. The old method, which actually won a Nobel Prize, is kind of like a sledgehammer. You're forcing brand new genes into a cell, basically wiping its hard drive and installing a new operating system. It works, but it's slow, it's inefficient, and frankly, it's pretty risky. This new idea, though, it's so much more elegant. It's based on the theory that the cell never actually deleted its youthful instructions. It just zipped them up and saved them in a hidden backup file. And the incredible part, scientists think they've just found the password. To really get this, we have to look away from the cell's head office, the nucleus, and zoom in on the factory floor, the cytoplasm, because floating around out there is this mysterious little structure that holds the key to this whole thing. It's called the P-body. So, what are P-bodies? Well, they're these dense little blobs just hanging out in the cell. For the longest time, science pretty much agreed they were just the cell's recycling centers. The idea was once a cell was done with a genetic blueprint, that's a piece of mRNA, it would just send it over to the P-body to get shredded. A simple biological junk drawer, right? Case closed. Well, this is where the story does a complete 180. It turns out the P-body isn't a junk drawer at all. It's a high-security vault. The cell doesn't destroy its old blueprints, especially not the really powerful ones from when it was young. Instead, it locks them away. It's not about forgetting the past. It's about actively repressing it. It's creating a cellular memory of what it used to be. So here's how it plays out. When a cell is brand new, it's what we call totipotent. All its blueprints are active and ready to go. It can build a whole person. But as it starts to specialize, let's say it decides to become a skin cell, it takes all those powerful youth blueprints, gathers them up, and sends them to the P-body. Then the P-body literally assembles around them, locking them away, silencing them, and creating this hidden memory of its past, more powerful self. And this, of course, led researchers to the next obvious question. If the P-body is a vault, then there has to be a lock. And if there's a lock, there has to be a key. This is where the scientific detective story gets really good. And that master lock, it turns out to be a single protein called DDX6. You can think of it like the keystone in an arch. It's the one protein that holds the entire P-body structure together. As long as DDX6 is doing its job, that vault stays shut, and all those amazing youth blueprints stay silent and locked inside. So the experiment they designed was just beautiful in its simplicity. Researchers used the CRISPR tool to go into cells and just turn down the DDX6 protein. They didn't add anything new, they didn't force anything in, they just broke the master lock that was already there. And what happened next was just astonishing. The P-bodies just dissolved. The vaults burst open and all those long-lost blueprints for a younger, more powerful state came flooding back out onto the factory floor. All of a sudden, the cell's machinery started reading instructions it hadn't seen in ages. The cell didn't need to be reformatted from the outside. It literally just rewound itself, accessing that latent file and snapping back to a more primitive, more powerful version of itself. And look, this isn't just some cool trick in a petri dish. It has immediate, powerful applications. Let's talk about infertility. Creating the precursors to sperm and eggs in the lab is notoriously difficult. The old way has a success rate under 8%, but by using this P-body method, by unlocking the cell's own hidden potential, that success rate jumped to over 24%. That's not a small step. That is a threefold leap in efficiency.
This one breakthrough points to two absolutely revolutionary futures. First, a real practical cure for certain kinds of infertility, making lab-grown gametes a real possibility for people. And second, it gives us the ability to create the true holy grail of stem cells, totipotent selves that can become not just any part of the body, but also the support stuff, like the placenta. This is huge. It could let us grow complete, complex organs for transplant someday. So we found the cellular rewind button. This all sounds amazing, right? A miracle cure for everything? Well, with great power comes great risk. And the catch with this technology, it's a massive one. This is a true double-edged sword. Before this technology goes anywhere, there are two huge, profound risks we have to face head-on. One is about pure biological safety, and the other, well, it's an ethical minefield that we are just not prepared for. First, the safety. That DDX6 protein we're messing with isn't just some random part. It's linked to really serious cancers like leukemia. In fact, the very process of blowing open the P-body is described as being almost identical to what happens when a cell turns cancerous. That is a terrifyingly high risk. Then there's the ethical side. With this tech, we can now create synthetic human embryo-like structures from skin cells that fall completely outside our current laws. We're stepping into an ethical Wild West with no map and no rules. Okay, so we have this incredibly powerful, but also incredibly dangerous new tool. The question now is, how do we make it safe? How do we refine it? The entire future of this field depends on moving from that blunt instrument to a precision tool. And that brings us right back to our core challenge. Right now, our method is a sledgehammer. We're suppressing all of DDX6. It's like blasting a safe with dynamite. Sure, you might get the gold inside, but you're probably going to blow up the whole building in the process. What we need is a scalpel. We need a way to carefully pick the lock and release only the specific blueprints we want, while leaving all the dangerous stuff safely locked away. And the cool thing is, we already have technologies that could get us there. We can use AI to design smart molecules that are programmed to only find and release one specific blueprint. Or we could adapt CRISPR, using a version called Cas13 that targets RNA instead of DNA to act like a tiny molecular hook that can go into the P-body vault and physically pull out only the one instruction manual we actually need. If we can figure that out, the long-term vision here is nothing short of transformative. It's called in vivo reprogramming. Imagine just getting an injection, a drug that travels to, say, a damaged part of your heart, temporarily picks the lock on those cells' P-bodies, releases the specific instructions for self-repair, and then it's gone, letting the vault lock right back up. No surgery, no transplants, just triggering our body's own hidden ability to heal itself. So this discovery is a genuine paradigm shift. We found this powerful and, yes, dangerous new tool that could completely rewrite medicine as we know it. The question for all of us now is, what do we do with this power? Is this cellular alchemy the key to a healthier future for humanity? Or are we opening Pandora's box to an ethical nightmare we just can't foresee?